Hello, and welcome to this overview for lab number 5 on muscle EMG and skeletal muscle function for Biology 206 Human Physiology. And so just a list of the objectives, and these are also on the lab report and lab handout. So the focus of this week's lab is to take a look at basically motor unit function, looking at muscle function and the control of muscles. And so to kind of review some of the concepts from lecture, keep in mind that what we're focusing in on are going to be these lower motor neurons. Motor neurons are going to be located within the ventral horn of the spinal cord. These cells are going to send a process out, send an axon out through the ventral root, down through the peripheral nerves, and these are going to be the named nerves that you would get in like an anatomy class, ultimately going down and making contact with the skeletal muscle that motor neuron is going to be controlling. Okay, so we're going to have the motor neuron, the axon process going out, and then as we get out into the periphery, into the actual skeletal muscle itself, we're going to see that these axons are going to be able to split, to branch out, so that they make connections with multiple skeletal muscle fibers. And so a muscle unit or a motor unit is going to be the motor neuron within the spinal cord, and then all of the muscle fibers that it innervates. So in this case, we've got kind of a reddish motor neuron coming here, making a connection with these, the first, the third, and the fourth of these muscle fibers. We've got kind of a more purplish one or maybe bluish one coming through. And again, that is making a connection with, say, the second and the fifth of these muscle fibers. And so what we can look at then is going to be the control of these skeletal muscle cells by these motor neurons, and then measuring skeletal muscle activity using the Power Lab and lab chart system. The first thing we're going to look at is going to be electromyography. And so basically what we've got with that is an idea that we are going to have a detecting electrode that's going to be located above one of our gross anatomical muscles, so, like the biceps or the triceps. We're going to put a reference electrode out there on the body, again, to serve as a point to, compare the amount of electrical activity, the amount of kind of ion flow that we're going to measure as our electromyograph or EMG, so that when the muscle is contracting, we're actually going to see the force of the contraction, the kind of electrical signals are going to be picked up by these electrodes. Now, in the first activity, the focus is going to be alternating between flexion and extension. And so with flexion, we're going to actually see the biceps muscle here contracting, the triceps as the antagonistic muscle is going to be relatively relaxed, so that when we have a contraction of the biceps, we're going to have flexion of the arm. When we have extension of the arm, we're basically going to see the biceps being relaxed and the triceps contracting. Now, the focus of the first activity is to look at co-activation of antagonistic muscles. And so, we describe this as, the biceps contracting and the triceps relaxing during the process of flexion. But we're also going to have a little bit of activity here within the triceps, this co-activation of the antagonistic muscle to kind of stabilize the growth, to stabilize the joint, to provide some level of muscle control on both sides of the joint being present here. So we're going to see a series, or actually an activity where you're alternating flexion, contraction of the biceps, and then extension, contraction of the triceps. In the second activity, we're going to be taking a look at the electrical stimulation of the nerve. And so we're going to use a stimulator. We are going to vary the amplitude of the stimulation and basically affect the number of axons that are going to be firing, or the axons that are stimulated by this electrical stimulation. Now, the number of axons that are firing is going to be related to the number of motor units that are going to be activated. Again, keep in mind that the motor units are going to be the motor neuron, the axons associated with it, and then all of the muscle cells that it innervates. So this week in Physiology Lab, what is it that we're actually going to be doing? And so like we've done throughout the semester, we are going to have the materials available for you on the Canvas module, module number 5. And so we've got the kind of detailed instructions from the manufacturer, ADI, the manufacturer of the Power Lab system, and the lab chart software. We've got a PDF version of the lab report template and additional instructions. PDF is provided in case you kind of edit the version that you've got and you need to go back and compare what was there originally. Now, the template for the lab report and instructions for the assignment this week are going to be available as a Google Doc. You're going to be able to download this either onto your computer if you're going to use Microsoft Word, or copy it into your own Google document if you're going to be using Google Doc. The first activity is going to be alternating activity, alternating the biceps contraction and the triceps contraction. We're going to measure the EMG amplitude on both the biceps and the triceps, and then compare them. In terms of additional resources, we have a data set for co-activation if you run into difficulties or are unable to attend the laboratory setting. In the second activity, we're going to look at twitch response, recruitment, and summation. And so basically, we're going to be measuring EMG amplitude in response to different levels of electrical stimulation of the median nerve. And so basically kind of control what is going to be the level of stimulation, the amount of activity, and then recruit kind of additional numbers of axons, additional numbers of motor units. Again, within the additional resources on Canvas, we've got a data file for the twitch response, which is available to you if you need it. Once you've completed the lab activities, answered the questions at the end of the lab report, you're going to submit your lab report, again, hopefully with the proper naming convention. So submit your lab report at the proper location within Canvas. As always, your lab instructor may have additional information available to you. And so please see if they have any changes or updates for the lab for this week. As always, if you have any questions, please do not hesitate to reach out to me. Thank you, and have a great week in lab.